Hello, this is Dr. Kudakasseril. This is the fourth video of the series of videos I am doing on biotechnology. Today I am going to talk about the steps in genetic engineering. The first step in genetic engineering is isolation of the gene. Usually we isolate genes from a cell culture. For example, if you are going to isolate a human insulin gene, you have to first make a human cell culture. Second step is called amplification of the gene. There we are making multiple copies of the gene. The old technique is called gene cloning in bacteria, where we use cloning vectors such as PBR322 and PUC18, details of which I am going to talk in the next video. Second technique is called polymerase chain reaction or PCR. There we use a machine called the thermal cycler. It is a very fast technique in which within 90 minutes we can make 1 billion copies of the gene. So it is a much better technique compared to gene cloning in bacteria. Third step is insertion of the gene into an organism. There are two methods. One is direct gene transfer that does not use a vector. For example, gene gun method does not use a vector. You have to remember one thing. Direct gene transfer is not preferred because it results in only 1% transformation. The second one is indirect gene transfer using a vector. For example, in plants, the vector used to transfer genes is the TI plasmid of Agrobacterium tumefaciens. Fourth step in genetic engineering is screening of transformed cells. There, what we are doing is using a marker gene, we identify the cells which contain the foreign gene. We can either use antibiotic resistance genes as marker genes, or you can use green fluorescence protein gene, which is obtained from jellyfish, as the marker gene, and luciferase gene from firefly as the marker gene. So these are the four steps in genetic engineering. Now let us look at the first step in genetic engineering, isolation of the gene. In this step, what we are doing first is we are isolating DNA from a cell. If you are isolating DNA from a plant cell, first you have to remove the cell wall using enzymes such as cellulase, hemicellulase, and pectinase. If you are isolating DNA from a fungal cell, you use chitinase. If it is a bacterial cell, we use lysozyme. Then using protease, we remove proteins in the cell. In the next step, using ribonuclease, we remove RNA. Then we add chilled ethanol or isopropanol when DNA precipitates. This precipitated DNA comes to the top. It is spooled on a glass rod. This is in the picture you can see the isolated DNA. The isolated DNA is then cut with a restriction endonuclease when hundreds of DNA fragments are obtained. Then we separate the DNA fragments based on their size and charge differences using a technique called gel electrophoresis. The details of gel electrophoresis I will discuss in the next video. Thank you for listening to my video. I have published some books for students appearing for the NEET exam. One is Handbook of Biology, Volume 1, which describes all the chapters in class 11, and Volume 2, which describes all the chapters in class 12. And also I have published a book, Biology in a Nutshell, which includes all the chapters. These books are available on Amazon. I am giving the link below. In the next class, I will talk to you about vectors and their use in genetic engineering. Thank you for listening to my video.